Welcome to the Chapter 22 video lecture on metabolism and energy balance. Please refer to the Chapter 22 study guide to see the learning objectives and the tables and figures that you'll be responsible for this chapter. Now, the control of food intake into our digestive system is a highly regulated and complex process. And we can attribute the two behavioral states that dictate whether we feel hungry or whether we feel full as the appetite center in the brain and the satiety center in the brain. And these two regions or these two behavioral states are under control by the hypothalamus. So the feeding center is what we call tonically active, and that means that typically our default state would be in the feeding center. That's what's normally being stimulated in the brain. The satiety center acts as an inhibitory pathway that inhibits that feeding center. So it kind of disrupts that tonically active control. So there's two theories for the regulation of our food intake. The first being the glucostatic theory, and this focuses on the hypothesis that glucose metabolism is primarily what is driving the hypothalamic feeding centers in our brain. The second theory is what we call the lipostatic theory. And in this theory, we're driving the feeding center by signals in our fat cells and adipose tissue. So as we start building our fat storage, we start secreting leptin, and then leptin has a negative feedback in which we turn off this notion of producing neuropeptide Y. Neuropeptide Y is a peptide that's being released in the brain that stimulates the hypothalamic feeding center. So if we turn this off, we actually stop our food intake. In addition, there's a lot of other neuropeptides and hormones that are being utilized in the lipostatic theory. Some of those hormones include CCK, glucagon-like peptide, PYY. All of these we talked about in the previous chapter are released in the small intestines when we bring food into that. So these two theories, it's not that one is correct and the other is false. There's evidence that both of these things drive regulation of food intake. At this point, let's define metabolism. When we talk about metabolism in the human body, we're really talking about the sum of all chemical reactions in the body. And we use those chemical reactions to extract energy from nutrients. We then can use that energy to do work, and that if we still have leftover energy, we can store that energy in biomolecules that can later be utilized for their high energy states. Metabolism can then kind of be categorized into two pathways. We either have the anabolic pathway, in which we are building large molecules from small molecules. So typically, we utilize anabolic pathways when we're in the fed state, meaning we have food, we have energy stored, we're in an absorptive state, and we can build larger molecules. So we can store energy as fat. We can make glycogen in our muscles. Those would be anabolic pathways. The opposite is what we call catabolic pathways, and this is when we're breaking large molecules down in the body into smaller ones so that we can utilize them for energy. Typically, we use catabolic pathways when we're in the fasted state or where we don't have food. It's a post-absorptive state. Now, when we talk about metabolism, it begs the question of how do we measure metabolism? How can we measure one's metabolic rate? Researchers have devised a number of methods to try to infer metabolic rate. Sometimes we get what's called a basal metabolic rate, in which we measure the lowest metabolic rate of an individual. We can do that after maybe fasting for 12 hours. Sometimes we call that a resting metabolic rate. So you do a 12-hour fast. We know we're in the fasted state, and then we try to measure what that metabolism would be in the given person. And we can do this in some different ways. We can try to measure or infer metabolic rate based off of oxygen consumption or CO2 production. There's a number of different ways that can be done. Now, some of the things that affect our metabolic rate, there's 6 factors that can affect what one's metabolic rate. First is going to be your age and your gender. So males and females have different metabolic rates, and as you get older, your metabolic rate tends to decrease. Second is going to be the amount of lean muscle mass that an individual has. Third would be their activity level. Fourth is going to be their diet and diet-induced thermogenesis. Fifth is going to be the hormones that an individual has. And sixth is going to be their overall genetics. This genetics plays a large role in some of this, thermogenesis, in the amount of lean muscle mass. Genetics plays a role in those. So if you look at this, really the only thing that we can control, we can't control our age and our gender. We can't control a lot of the amount of lean muscle mass that we have. That's largely genetic. We can't really control our hormones. We can't control our genetics. So we really only have control over our activity level and our energy intake, so that our diet. Those are the only 2 things that we can really voluntarily change out of those 6 factors. So when we eat food, what happens to that food? And how does it then become energy? Well, as we ingest biomolecules, they have 3 fates. They can either be broken down into energy to do mechanical work. We can break them down and then use them for the synthesis of growth and maintenance of the body. And then lastly, we can store that energy as either glycogen or fat. Okay, so this is, the basic amount of energy of energy we need to just maintain ourselves, to grow and maintain. If we have excess, we can maybe put a little bit more into growth, but we have certainly a maintenance function. Then if we have additional energy, we can use that to do work, apply it into growth, or we can put it into storage. This brings us to the concept of nutrient pools. So in the body, we have nutrient pools that contain nutrients that are immediately available for use. And we can put those into 3 pools. So we have what's called the free fatty acid pool, in which we have fatty acids. We have the glucose pool, and we have our amino acid pool. Now this figure is taken from your book, and it's figure 22.3. This figure summarizes your entire body's metabolism and how we move nutrients from free pools into storage pools. And you need to make sure that you understand this figure and the processes that get utilized to move nutrients from storage into free access pools. So let's go through this together. Let's start with fats. We ingest fats, we digest them, we break them down into fatty acids, so free fatty acids and glycerol. We'll take that glycerol and we'll utilize it for metabolism in some tissues, and then we'll take the fatty acid and we'll put it into our fatty acid free pool. So these are now, we can use fatty acid and we can take those fatty acids and immediately metabolize them if needed for energy. And the way that we would do that, the pathway, and it's not written on here, but the pathway in which we would utilize fatty acids and convert it straight into energy is called beta oxidation. So beta oxidation is how we get ATP through aerobic pathways and utilize it for energy. Now, if we want to take that free fatty acid and we want to store it, then we are going to use a biochemical process known as lipogenesis, so fat lipogenesis and put it into adipose tissue and fat storage. Okay? If we need to convert that fat back into fatty acids, we would use lipolysis. So again, fat lysis is breaking apart, so breaking apart fats. So lipogenesis stores, fat, lipolysis degrades, and beta oxidation turns it into ATP. Now let's move to carbohydrates. So when carbohydrates we digest, we absorb, and we're going to break carbohydrates down into glucose. Okay, glucose is then going to be moved into our glucose pool. All right, if we have excess glucose, it might be excreted. But let's just say this is our normal range of plasma glucose. And we can then use glucose for metabolism through glycolysis. Okay? So glycolysis is going to be the process by which we turn glucose into ATP. You guys learned about that in Bio 112. It's respiration, cellular respiration, utilizing glycolysis, the Krebs cycle, okay, electron transport chain. All those things will release the ATP out of glucose. If we want to store glucose, we'll store it as a molecule known as glycogen. Okay, and we turn glucose into glycogen through glycogenesis. Okay, so you can then think of, like in this term, glycogenesis, right? So glycogenesis, we're creating glycogen. We break glycogen back down into glucose through glycogenolysis. Okay, so glycogen lysis. There we go, breaking it down. So just to recap again, glucose can be utilized through glycolysis in the Krebs cycle and electron transport chain. We can store it using glycogenesis, and we can break it down through glycogenolysis. Now let's look at proteins. So proteins get ingested, digested, broken down into amino acids that'll be then moved into the amino acid pool. Amino acids can be used as energy by converting them into glucose. So we do that through gluconeogenesis. Okay, that's how we take amino acids through gluconeogenesis and convert them into glucose. We can store our amino acids by converting it into protein. So we utilize protein synthesis. Now in the liver, we can also turn proteins immediately into energy using deamination. So deamination is how we would immediately use it for metabolism in the liver. Now you may have noticed here we talked about how we could take amino acids and move it into the glucose pool. We can take glucose, we can also move it into fat. We can take excess glucose and move it over into the fat storage. We can do that through lipogenesis as well. So we can kind of move things back and forth a little bit through these different pathways. This table is taken from your book. Again, it's a great table that kind of breaks down what happens to carbohydrates, proteins, and fats in our two states. So whether we are in the fed state of metabolism or whether we're in the fasted state of metabolism. And it just outlines what I went through on that previous figure. Okay, in the fed state, glucose can be used immediately to get energy by using glycolysis in the citric acid cycle. We can store it as a lipoprotein in the liver. We can store it as glycogen in the liver and in our muscle cells through glycogenesis. We can convert it to fat through lipogenesis. Okay, if we haven't eaten, we're in the fed state, then what's going to happen is we're going to be breaking down glycogen through glycogenolysis to get glucose so that we can utilize that in the liver, in the kidney, in our muscles for glycolysis. Proteins, likewise, if we are in the fed state, most amino acids are going to go to the tissues for protein synthesis. That's typically what we're going to utilize it for. We're going to build more proteins. If we do need energy and we don't have glucose available, then we can convert those amino acids in the liver to intermediates that they can be utilized for aerobic metabolism and glycolysis in citric acid through deamination. And if we have excess amino acids, then we can convert it to fat through lipogenesis. If we're in the fasted state, we break down proteins into amino acids, and we would do that through a series of biochemical pathways. You don't need to know all of them. They get kind of complex depending on what type of protein we're breaking down. But we can then deaminate them in the liver for ATP, or we can use them to turn it into glucose through gluconeogenesis. Fats, their primary use in the fed state is going to be converted into adipose tissue through lipogenesis. We're going to store them as fat. Primarily that fat gets stored in the liver. We can also utilize them in the fed state to make cholesterol for our steroid synthesis, and we can use fatty acids for additional lipoproteins and some other compounds that get synthesized. In the fasted state, our triglycerides are broken down into fatty acids in glycerol using lipolysis, and then we can convert fatty acids into ATP by using beta oxidation. This is just kind of summarizing where some of the stuff gets stored and broken down in the body. So in the fasted state, metabolism is utilized to maintain concentrations of plasma glucose so that the brain can function. And where we see that primarily is in the liver. This is where we store most of our glycogen. Most glycogen gets stored in the liver. The other area where glycogen can be stored is in our muscle cells. So in the liver, we're going to start breaking down that glycogen using glycogenolysis to create energy and glucose. We can then use that glucose to supply the brain with energy. The fat that's been stored in the brain can get broken down into ketone bodies through beta oxidation, and we can use ketones to actually nourish the brain and turn it into ATP. The second place, so that's the first area we're going to go to. If we still have energy needs, we're going to move into our fat cells, in which we're going to break up the triglycerides into fatty acids using lipolysis, and then we'll convert it into energy production once we have glucose, and utilize those in the muscle cells to create, our ATP. In the muscles, glycogen gets broken down into either pyruvate, or if we're in an anaerobic state, lactate. And then we use gluconeogenesis to transport that glucose into the liver and then into the brain to be utilized there. Now, I mentioned there was two hypotheses that kind of drive our homeotic control of metabolism, the lipostatic and the glucostatic. We're going to focus now on the glucostatic, meaning the glucose plays a large role in it. And to understand that homeostatic control, we are going to look at what happens and what directs that glucose storage in the body. In the pancreas, we have endocrine cells that secrete hormones. There's two types of cells. We have alpha cells and we have beta cells. And alpha cells, they produce a hormone called glucagon. And beta cells produce the hormone insulin. And metabolism, specifically glucose metabolism, is controlled primarily by a ratio of these two hormones. Do we have more insulin than glucagon, or more glucagon than insulin? In the fed state, when we have food readily available and blood plasma levels, glucose plasma levels are high, insulin is going to dominate. That's going to be the predominant hormone that's driving our metabolism. In the fasted state, when our blood glucose plasma concentrations are low, glucagon is going to dominate. So let's look at insulin and what factors play a role in insulin secretion. So the primary factor is going to be an increase in plasma glucose concentrations. So when the blood plasma glucose concentrations is greater than 100 milligrams per deciliter of glucose, that's going to stimulate the beta cells in the pancreas to secrete insulin. Okay, that's the primary. Additionally, increased plasma amino acid levels will also stimulate the beta cells to release insulin. And then from our lipostatic theory, there's also these feed-forward effects of all of our gastrointestinal hormones that we talked about last time. So we now know that almost 50% of our insulin gets secreted by the glucagon-like peptide 1. That's what stimulates it. This is produced in the ileum, in the jejunum, as we start to fill and absorb food in them. We trigger the release of these gastrointestinal hormones. And the purpose for that, for this feed-forward control, is we already start secreting insulin before that glucose reaches the plasma, which ensures that we can utilize it a little bit faster and that we don't have as rapid of spikes in blood plasma glucose levels. Additionally, the parasympathetic activity of the autonomic system, so if we have parasympathetic innervation of the pancreas, that will trigger those beta cells to release additional insulin, and the sympathetic activity would inhibit it. Okay, so these are antagonistic. So what does insulin do exactly? The primary function of insulin is to lower plasma glucose concentrations. And we do that in a number of ways. First way is by increasing glucose transport into what we call insulin-sensitive cells. And we can do that in a couple of ways. Let's look at two types of tissues that have insulin receptors. So here is, this would represent either fat, so adipose tissue, or skeletal muscle. They have insulin receptors. When insulin is not present, glucose is not necessarily going to be readily absorbed into these two types of tissues. So in the fasted state, glucose doesn't enter adipose tissue, doesn't enter our skeletal muscles easily. In the fed state, what happens is insulin will bind to insulin receptors. That triggers a signal transduction cascade of a lot of different proteins being turned on and produced. But the short story is that it triggers exocytosis of secretory vesicles that contain GLUT4 transporters. And so now we have GLUT4 transporters that get put into the cellular membranes, and this allows glucose to then move into our skeletal muscle cells and into our adipose tissue. In the liver, things are a little bit different. So the liver, it always has GLUT2 transporters. Remember, the liver is the primary spot that we're going to store glucose. So even in the fasted state, glucose may be moving in through, or glucose is going to be able to move through these transporters. However, GLUT2, again, it's a passive transporter, just like the GLUT4. So we're moving from high to low, and during the fasted state, we're going to have high concentration in the liver, low concentrations in the plasma. So we'll actually be moving glucose out of the liver and into our plasma. What happens during the fed state, not only do these ratios change to where we now have higher glucose outside, so it'll naturally be moving in, but when insulin binds to the insulin receptors, the signal cascade, it triggers the activity of hexokinase to phosphorylate glucose into glucose 6-phosphate, which can be utilized in the Krebs cycle. So it's essentially, it's activating glycolysis and initiating the Krebs cycle so that we can extract ATP. And the beauty of this is as we convert glucose into glucose 6-phosphate, the intracellular glucose concentration stays low so that we can bring in more glucose into the liver cell, and so we're reducing our plasma glucose concentration levels. Okay, so function was to increase glucose transport. Function 2 is to enhance cellular utilization and storage of glucose. So at the same time that we're moving glucose in, when we have these receptors bound, it's also going to activate enzymes for glycogen synthesis. Glycolysis, glycogenesis can occur. We're also going to inhibit the enzymes that would normally be breaking down glycogen. So glycogenolysis, gluconeogenesis, lipolysis, all those processes are going to be inhibited and will stimulate glycolysis and glycogenesis. Third, we're going to enhance the utilization of amino acids. So again, remember, insulin gets released at high amino acid concentrations, not just glucose concentrations. And this activates the enzymes when insulin binds to these receptors inside those cells. It's going to activate the enzymes for protein synthesis and inhibit enzymes for protein breakdown. And lastly, insulin, when it binds to insulin receptors, is going to stimulate or promote fat synthesis. So we inhibit the enzymes for beta oxidation, and we promote the conversion of excess glucose and amino acids into triglycerides. So we promote lipogenesis. Therefore, we call insulin an anabolic hormone. So just to recap, let's look at all of the functions that occur when we eat a meal. So we eat a meal, two things are going to happen. One, we're going to now have presence of carbohydrates in the GI of our lumen. Okay, that's going to trigger endocrine cells in the small intestines to start producing that glucagon like protein 1 and GIP. Those will stimulate the beta cells of the pancreas to increase insulin production. At the same time, we're going to have the GI tract is going to have distension. It's going to start expanding. That's going to trigger stretch receptors that send a signal to the central nervous system that then trigger the parasympathetic system. So a parasympathetic neuron will stimulate the beta cells of the pancreas to start secreting and producing insulin. As we start absorbing, okay, these both are going to be kind of feed-forward controls. But as we start absorbing now, the amino acids and the glucose, that's going to cause a spike in our blood glucose concentration. That increase in blood glucose concentration and amino acid, that also triggers the release of insulin from our beta cells. Additionally, the increase of plasma glucose concentrations has an inhibitory effect on the alpha cells of the pancreas. So we're going to stop releasing or secreting glucagon. Now, as our liver increases, we're going to have effects on the liver, as our insulin increases, we're going to have effects on the liver and on our muscle and adipose tissue. And what we'll see in the liver is that we're going to increase these biochemical pathways. So we're going to increase glycolysis, we're going to increase glycogenesis, we're going to increase lipogenesis, we're going to increase protein synthesis. So these are all anabolic pathways. In our muscle and adipose tissue, we're going to increase the glucose transport. That occurs also in the liver. And inside muscle and adipose tissue, we'll also be doing these anabolic processes. And as we do these, and as we increase glucose transport, our plasma glucose concentration is going to drop. And as it drops, it has a negative feedback loop, right, on our plasma glucose concentration. And that negative feedback loop can then alter whether we're utilizing the alpha cells or the beta cells. So we talked about insulin. Now let's talk about glucagon. So glucagon is antagonistic. Okay, again, remember, these kind of have the opposite effects. So when glucagon is present, we're going to be using glycogenolysis. We're going to be stimulating gluconeogenesis, and we're going to be stimulating ketogenesis. It gets secreted by the alpha cells, and it functions to prevent hypoglycemia, so too low of blood plasma glucose concentrations. So as soon as we get below that 100 milligrams per deciliter, we're going to really start ramping up the secretion of glucagon. The primary target tissue of glucagon is going to be the liver. That's where we're going to have most of the glucagon receptors. So let's look at what this looks like. Blood plasma glucose concentrations drop. When that drops, we're going to then stimulate the alpha cells of the pancreas to produce more glucagon, and we will inhibit the beta cells of the pancreas. So we're going to stop insulin production. As we stop that insulin production, what that means in our muscle and adipose tissues is that we start metabolizing and producing lactate, pyruvate, amino acids, fatty acids. We start breaking down. We're using catabolic pathways that then move those substances through the blood into the liver, and in the liver, we will then be stimulating, because of glucagon binding to receptors, we will stimulate the catabolic pathways of glycogenolysis, of breaking down glycogen into glucose, gluconeogenesis, of taking amino acids and pyruvate and converting it into glucose, and then fatty acids, we will, if we have a prolonged hypoglycemia, and so we're just not able to generate glucose to increase this plasma glucose concentration, the brain can utilize ketones and derive them as an energy source. So as an last-ditch effort, we'll start using ketogenesis to take those fatty acids and turn them into ketones so the brain and some peripheral tissues can get some amount of ATP. The last part that the book talks about in this chapter is conditions like diabetes, and what happens if you don't have insulin being released. How does that play out in the body, and what problems do we have? Now, I'm not testing over these things, but I want to go through this to explain how important insulin production and insulin binding to these receptors really is. Because when we don't have this, a lot of things can go wrong. So let's look at our three types of metabolism: fat, glucose, and protein. And when we eat a meal, we absorb fatty acids, glucose, amino acids are increased in our blood plasma. But if insulin isn't released, if we're diabetic, and let's say we have type 1 diabetes in which the beta cells are actually destroyed, and so there is no insulin release, well, then what happens is, instead of storing fat, the body still thinks it needs to break down fat. And as we break down fat, we further increase our plasma fatty acid concentration. We then start to have tissue loss over in our fats. In our plasma, amino acids, amino acid uptake by most cells decreases, and we start breaking down proteins. Even though we have plenty of this, the body doesn't know that. It hasn't been triggered. So we further increase our plasma amino acid levels. And we start to utilize proteins to generate our ATP, because we're not able to transport the glucose that is in their blood into the cells. It remains in the blood and not in our cells. So what do these things cause for our glucose metabolism? Well, glucose uptake is really low because those cells have not been triggered by insulin to bring glucose into the muscle and adipose tissue. Our glucose utilization remains low, and in the liver, we're still producing those catabolic enzymes, which leads us to become even more hyperglycemic, meaning we have a high glucose concentration in our blood. Likewise, because we're not able to utilize glucose, the brain starts to interpret what's going on, the fact that the cells aren't generating ATP, as starvation. And so we enter this phase of what we call polyphasia, which the brain is triggering the hypothalamus to go into the fed state. It's saying, hey, you need to eat. That causes additional dehydration. Okay? How does dehydration start to occur? Well, okay, as we're hyperglycemic, what that means is in our kidneys, we exceed the renal threshold for glucose. Too much glucose, we can't reabsorb it. And so we get what's called glucosuria, in which our urine is high in glucose. That causes osmotic diuresis and polyuria, in which now we start to become dehydrated, because as we move glucose out into the lumen, water is going to move with that too. So we're urinating. That's what diuresis and polyuria is frequent urination. Our blood now becomes, the osmolarity is going to start to increase. Okay? Why does it start to increase? Because we have a lot of glucose in there. So water is going to be moving out of the cells into the blood to try to balance out that. As that happens, that triggers the brain and says, like, hey, we're becoming dehydrated. We need to drink more. So we increase the production of our ADH hormones. We increase thirst. That's called polydipsia. Okay, so the brain's saying, hey, we're thirsty. We need to drink more. If we try to compensate for that, but if dehydration continues, then guess what? Our blood volume and our blood pressure at some point starts to drop, because we cannot compensate by pulling more and more water out of the cells. The body then tries to deal with this by increasing the heart rate, right? Increasing the stroke volume as little as it can through the cardiovascular control center. And what that ultimately leads to is circulatory failure. The heart can't keep going. And when we have circulatory failure, we enter into a diabetic coma. So that's one half. That's just on a water aspect. The fact that our solute concentrations in the blood get thrown off because of the glucosuria. At the same time, as we start utilizing ketones for ATP, because we can't get that glucose into the cells and the brain needs to survive, we start producing more and more ketones for brain metabolism. That causes ketoacidosis, which changes the pH of our blood. And as that happens, we get into what's called metabolic acidosis, in which now our pH is dropping, and that triggers, again, our central chemoreceptors to recognize the fact that, hey, our pH is off. So we start increasing our ventilation. We start breathing rapidly to try to get rid of, what it assumes is CO2, right? Our urine becomes acidic, and we start having a lot of excess potassium that is being that's now in our blood and moving into our urine. So we call that hyperkalemic. Likewise, as we start increasing the circulatory center, we start working in the heart harder and harder. At some point, we move into anaerobic metabolism, which creates lactic acid, which contributes further to that metabolic acidosis. And just from a pH side and a ventilation side, this puts strain on the body and can lead to coma or death. Now, I'm not testing over all this. I put this in to help bring this all together to show how important insulin can be, because if you can't take glucose in, all of these pathologies can arise.